A region of interest, or ROI analysis, is a method of extracting data from a subset of voxels, also called a mask. One way to create a mask is to use an atlas, or a map that partitions the brain into anatomically distinct regions. Another technique is called the spherical ROI approach, in which a sphere of a given radius is centered at a triplet of specified x, y, and z coordinates. These coordinates are often based on the peak activation of another study that uses the same or a similar experimental design to what you are using. In this tutorial, you will learn how to do both types of ROI analysis. FSL has many atlases already installed, which you can access through Fossilize. First, load the MNI template that you normalized your data to. Then, click on Settings, OrthoView 1, and select the Atlas panel. It will open up a new window called Atlases. By default, the Harvard-Oxford cortical and subcortical atlases will be loaded. You can see how the atlas partitions the brain by clicking on the Show Hide link next to the atlas name. The Harvard-Oxford atlas is a probabilistic atlas, which means that the voxel at the center of the crosshairs in the viewing window is assigned a probability of belonging to a brain structure. To save one of these regions as a mask, click on the Show Hide link next to the region you want. In our example, let's say we want to use the paracingulate gyrus. Clicking on the link will show that region overlaid on the brain, as well as load it as an overlay in the Overlay List window. Click on the disk icon next to the image to save it as a mask. Save it to the flanker directory and call it PCG. Once you've created the mask, you can then extract each subject's contrast estimates from it. For the incongruent minus congruent contrast estimate, for example, you can find each subject's data maps in the directory flanker second-level.gfeat cope3.feat stats. My preference is to extract data from the Z statistic maps, since they are standardized and easy to interpret. We will need to merge all of the Z statistic maps into a single data set. To do this, we will use a combination of FSL commands and Unix commands. For more details about how this command works, see the link for the book chapter in the More Info box down below. Move the allzstats.nai.gz file up 3 levels so that it's in the main flanker directory. Then use the FSL meanTS command to extract data from the PCG mask that we just created. This will print 26 numbers, 1 per subject. Each number is the contrast estimate for that subject, averaged across all of the voxels in the mask. You can then copy and paste these numbers into a statistical software package, such as R, and run a one-sample t-test to see whether the contrast estimates are significantly different from 0. These results, as you can see, aren't statistically significant. This may be because the anatomical mask is so large that it encompasses several distinct functional regions. In this case, we can instead use a spherical ROI analysis. For example, let's say that we read a paper examining cognitive control effects and that they reported a peak activation at the MNI coordinates 0, 22, 40. Now, the procedure in FSL for creating a sphere from these coordinates is a little weird, but stay with me. We first need to convert these MNI coordinates to voxel space, which can be found in Fossilize. Enter the MNI coordinates in these fields down in this window here, and notice how the numbers change over here, and then write down the numbers in the voxel location fields. In a terminal, type the following command. This is a long, dense command, but for now, just note where we have inserted the numbers 45, 73, and 58. What this command does is create a single voxel that we will use as a base for the next command. This command now expands the single voxel into a sphere with the radius of 5 millimeters and labels the resulting sphere as Jan sphere DMPFC 02044 to indicate the coordinates. Next, type this command, which will binarize the sphere so that it can be read by the FSL commands. This code is all pasted in the More info box down below for your reference. Load this mask into the fossilized viewer to make sure it is where you think it should be. You can hide the other layers to make it easier to see where the mask is. Lastly, we will extract data from this ROI by typing the following command. You can then copy and paste these numbers into R and run a one-sample t-test. Because this ROI is more localized, we have more power to detect the cognitive control effect. And with that, you have completed the fMRI short course. In the fMRI tutorial you just completed, you learned that group-level contrast maps are created through something called a mass univariate analysis. In other words, we carry out as many statistical tests as there are voxels. Given that a typical fMRI dataset contains tens of thousands of voxels, this can quickly lead to an unacceptably large number of false positives. To control for the number of false positives, therefore, and to keep them at the conventional false positive rate of 5%, we need to do something called multiple comparisons correction. In the good old days, many neuroimaging researchers used a correction method called Bonferroni correction. It's simple to understand and simple to do. Take your alpha level, or the false positive rate you're willing to live with, traditionally set at 5%, and divide it by the number of tests that you carry out. This works well enough for behavioral studies, but quickly becomes unreasonable when applied to imaging data. For example, if your group-level contrast map contains 100,000 voxels and your alpha level is 0.05, an individual voxel will have to pass a significance threshold of 0.00000005 in order to be judged statistically significant. Although Bonferroni correction will ensure that your false positive rate is no higher than 5%, it's also a very conservative test. Bonferroni correction is valid only when each test is independent. To take our fMRI dataset again as an example, that would mean each voxel is completely independent of every other voxel in the brain. Knowing the value of one voxel doesn't tell you anything about any other voxel. But are the voxels completely independent? Let's take a look at a typical fMRI image. If we zoom in, notice that a given voxel is similar to its neighbors. Bright voxels tend to be surrounded by brighter voxels, and darker voxels tend to be surrounded by darker voxels. Since we can make a reasonably accurate guess about what the signal intensity will look like for a given voxel, given its neighbors, the voxels are not completely independent. Bonferroni correction, then, is too severe. Although it virtually guarantees to keep your false positive rate below 5%, it will also probably lead to a high false negative rate. That is, you'll probably conclude that no effect exists when there actually is one. An alternative is cluster correction, which is the most popular correction method in fMRI analysis. Cluster correction takes advantage of the fact that the voxels in a typical dataset are not completely independent. Instead of testing each voxel individually, clusters of voxels are tested for significance. To illustrate this, let's take a coronal slice from the group-level incongruent minus congruent contrast you created in the fMRI tutorial. Notice how the brighter colors can be grouped into distinct clusters. If we look at this from a different angle, we can think of the clusters of voxels as mountain ranges. The height of an individual voxel is determined by its z-value, and higher z-values lead to higher peaks. The threshold that we apply is a cross-section through the mountains at a certain height. For example, a z-value of 3.1, corresponding to a p-value of 0.001, and we only observe the peaks that remain after applying this threshold. This is known as thresholding the image, or more specifically, setting a cluster-defining threshold, since only those voxels that are at or above that threshold will remain. And this threshold is the value that you see in the Post-Stats tab of the FEAT GUI. You may think that's all we need to do, and as far as entering numbers into the FEAT GUI, it is. But there's an additional hidden step happening after you press the Go button that you should know about. Remember that a cluster-defining threshold is not the same as an alpha level. It could be that the clusters as large as the ones in our group analysis are just as likely to be found in images created from pure noise. Let's say that our cluster in the medial prefrontal cortex is 50 voxels large. What we need to ask ourselves now is how often would we expect a cluster of that size to appear due to chance? To answer this, we run simulations. In other words, we create artificial data sets with the same dimensions and smoothness as our task data set, but which are composed of pure noise. We then write down the size of the largest cluster that passes our cluster-defining threshold, and repeat the process with another simulated data set. If we do this thousands and thousands of times, we can create a distribution of maximum cluster sizes. And from this distribution, we can calculate the percentage of the time we would observe a cluster as large as the one we generated from our task dataset. If that percentage is lower than our alpha level of 5%, we can reject the null hypothesis for that cluster. And these are the clusters that are automatically generated by FSL and stored in the images with the thresh prefix.